Hello everyone, welcome to a new video. So I'm doing this a little bit differently, got some new equipment set up here, so you'll be able to see my face now every time I teach, and I'm going to be able to draw a little bit more in real time, and hopefully this works out better. So last time we were together, we talked about bruising in this lecture. So what we said was this was the breakdown of hemoglobin. So now we want to talk about his blood types. Now in blood types, we need to get a few terminologies down. First, the two ones are going to be antigens and antibodies. So we got a combination of two things, antigen, antibody. What is an antigen? An antigen is really at first, as simple as I can put it, is this is anything that causes an immune response. Now, so an antigen is something on the surface of a cell, normally a foreign cell, but it could be present on normal cells as well, and it only forms an immune response if, and only if, we have antibodies that are against it present. And the antibodies attack antigens, so that means I've got to get antigen, antibody together in the same thing. So an antibody only attacks when there is an antigen. And what it does is, you can see here, where the antigens on the red blood cells and their opposing antibodies are present, they will cause a clumping and ultimately result in hemolysis. This is referred to as an agglutination. And one's blood type, though determined by DNA, by genetics, by one's genes, determines the presence or absence of certain antigens. Now, the ones we're going to talk about is the A, B antigens and the Rh antigen, but there are others. Now, to do this, what I do want to talk about is the blood types. I'm going to go to this first, and we're going to talk about some blood types, and we're going to start off here with my blood types. We have type A, type B, type AB, and then type O. So each one of these will have a red blood cell. Let me draw a circle for a red blood cell here. And these will be my red blood cells, RBCs, that we have. And these red blood cells will have antigens on their surface. Now, A has an antigen, and I'm going to use a triangle to draw it. It doesn't matter which shape we use, but these are called the A antigens. Now, there are particular genes that determine which ones go on that cell. So a person who has type A blood has only A antigens. Now, that might also go to, say, a person who has type B blood would only have B antigens on its cell surface. So here we have B antigen as well. Now, the B antigens that are present here on the cell surface for type B. Now, AB might tell you that it has both A and B antigen. So let's draw an A here and an A here, and then a B here and a B here. So they have both A and B antigen, both present. Now, type O will have no antigens present, hence type O. It has 0 antigens. Now, that is one thing. But the next thing would be the antibodies. So, antibodies, antigens. So what antibodies would there be? Now, here's the kind of breakdown. If I've got A antigen, the last thing I would want is antibodies against, or opposing antibodies, in my own plasma. So they are found in plasma of blood. And what we would see is you would not want to have antibodies against your own antigens. So they would have an antibody present called anti-B antibody. B blood, you would not want anti-B, so their antibodies in their bloodstream would be anti-A antibodies. Now here in AB, they have none. they have neither anti-A or anti-B. If they had anti-A, it would attack their A antigens. If they had anti-B, it would attack their B antigens, so they can have no antibodies present in that blood. But in O, since they have no antigen A or B, they will have both anti-A and anti-B present, both in the plasma. So these are antibodies, so they would have both anti-A and anti-B. Now, this isn't what the antibodies actually look like. Y-shaped protein is usually given as an antibody, but these antibodies we're going to see later on are different shapes than this, okay? So, with this sketch out of the way here, let's look at this on our notes now and see that what you've got is type A has the anti-A has the A antigen and anti-B antibodies. Type B blood has the B antigens with anti-A antibodies. AB blood has both A and B, but has no antibodies, and O has no antigens, but both anti-A and anti-B antibodies in the plasma. Now, given that, this creates a unique set of blood types. Now, the thing is, about 40% of the population is type A, 10% type B, 4% AB, and about 46% is O. Now, with that, these anti-A, anti-B antibodies are present at birth, but they're in very low amounts. Unless I get the wrong blood, if I'm type A and somebody gives me type B blood, then the antibodies that are anti-B will start to rise pretty quickly. But normally they're not there. Now, here's the deal. You might be wondering, well, how in the world can O be a universal donor when it's got both A and B antibodies? Well, what we do is we take your blood. When we donate your blood, you give your blood, they take that blood you donate, and it goes to the blood bank. The blood bank removes these antibodies, and as it removes the antibodies, you no longer have the cross-match issue with O. But it's when those blood cells are placed into the plasma that you can't remove the patient or recipient antibodies. So the antigens from the donor will interact with the antibodies of recipient. Okay? So this is where you get that cross-matching compatibility. Now, the third antigen called Rh, the most common one is called the D antigen sometimes, but there's two types. There's Rh positive, Rh negative. The positive means it is positive for the Rh antigen. The negative means it doesn't have it. So when you see the positive and negative, positive means it does have Rh, negative means it does not have Rh. Now, here's the thing about this. Antibodies against Rh, you don't even make those unless you get them. Now, why is this? Well, the antibodies against Rh are a type of antibody that you make. The same way you make those is the same way you make antibodies against the cold. So you have to get the cold virus in order to make antibodies against the cold virus. Same way you make anti-Rh antibodies. So if you are Rh negative and you get exposed to Rh blood, then that means you will make anti-Rh antibodies. So that means only a person who's Rh negative, who has been exposed to Rh positive blood previously, can make anti-Rh antibodies. Or they have been, we say, sensitized. Okay? Important. Now, this one, because these are made the same way you make antibodies against like a virus or bacterial infection, they make a lot of them and they can kill you. These actually, wrong blood type here, can cause severe reactions, even more so than will AB incompatibilities. So they tend to be a little more dangerous. So O negative blood is called the universal donor because there's no surface antigens on that surface and the antibodies have been removed. AB is universal acceptor because they have no antibodies present. AB positive can have no antibodies because they can't even be sensitized to have anti-AB. So they are the universal recipient or acceptor. We also call them universal plasma donor. Okay. Now, this is a case where these cross matches, what happens is when we try to make them non-compatible. So how does this happen? Well, we get what's called an agglutinogen. Your antigens, your blood type, is referred to as an agglutinogen. That is your blood type. If you're type A, then your agglutinogen is A. If you're type B, then your agglutinogen is B. If you're type AB, then you have both agglutinogens. If you have the O, then you have no agglutinogens. These are ignored by your own immune system because they're your own antigens. The agglutinins are what we call the antibodies. Your antibodies of your plasma. Agglutination is when your agglutinins come in contact with foreign, incompatible, or opposing antigens or agglutinogens, and they will clump and agglutinate, but it's called compatible if they do not do this, if they do not cross-react. This is if they don't form an agglutination. And that's very important for blood transfusion. So let's look at this. So this is something I will test you on this exam. So let's definitely hit that. And to do this, I'm going to draw 1, 2, 3, 4 cells. A, B, A, B, O. Okay, so with that, remember A has, and I just used triangles for A because they look like A's to me. Those are A's. B's, I use circle. It doesn't matter which symbol we use. It is just the fact, and AB has both. Okay, and O has none. Remember, the antibodies are also present. A has anti-B. B has anti-A. AB has none. And O has both. Okay, so what we're going to do with this drawing is we're going to draw a line to all compatibles. Let's get that autofocus there. There we go. Okay, so we're going to start off with A. A, blood. So here, I'm going to start drawing from the donor to recipient. So in donor, it's blood cell. In recipient, it's plasma. If A blood with A antigen was placed in anti-A plasma, it would be incompatible. Now, A blood, who has A antigen, goes into this. So AB, I can receive blood from A. A can go to AB. Why? There are no antibodies in the blood plasma to attack his antigen. Now, can A give to O? O has both. So it has anti-A, so it cannot. And A can then give to its self. So A can give to itself and AB, but A cannot give to B or O. A cannot give to B because B blood has anti-A antibodies in it. Okay. Now, let's start with B. Can B give to A? B has the B antigens. A blood has anti-B antibodies, so it cannot. But B can go to AB because there are no antibodies to attack the B antigens. B cannot give to O because it has both anti-A and anti-B, so the anti-B antibodies would take the B antigen of the plasma from the O patient, so that can't give. And B can then give to its self. So B can give to its self here. Okay? Now, AB. Let's look at AB. AB has both A and B antigens. If I try to give AB blood to A, the anti-B would attack the B antigens. If I try to take AB and give it to B, the anti-A antibodies would attack the A antigen, so it's not compatible. A here cannot give to O because O has both anti-A and anti-B, so he's in trouble both ways. So AB can only give to himself. Okay? Let's look at O. O has no surface antigens. If we took and tried to give it to A, we would be fine because A has anti-B, but there is no B antigen. O can then give to B because B here has anti-A antibodies, but there is no A antigens on the surface. And remember, when donating blood, the donor we've removed his antibodies from his blood and put it into the recipient's blood. So it is the donor's antigens and the recipient's antibodies that we're concerned with. O can also give to AB because AB has no antibodies for it to even worry about. Then O can give to itself. So AB can receive from everyone, O can give to everyone, and these are your cross matches. And if you draw this out repeatedly and make sure you understand what's going on, you can get this pretty well. And I'm going to ask you who can give to who on the exam. My exam will have who can give blood to who. Okay? Okay, now, and here you can actually see this is a serological test done in a lab. And in this lab here, you have, so here we have anti-A antibodies placed in a well, anti-B antibodies placed in a well, and anti-D or Rh, because D is the most common Rh antigen type. So that's also Rh. So you can see if somebody is A positive, that if A positive blood went into a well with anti-A antibodies, it clumps. But when it goes into a well with anti-B, it doesn't. And somebody who has A positive, who has a positive, clumps in anti-D because D is the most common Rh. So it means it is got antibodies against the positive Rh. Now with B, you could see that B blood, remember, has, so here it's not clumping, but here anti-B, it got attacked. Anti-D, it got attacked. AB, positive, attacked all across the board. O negative, no attacks across the board. Okay? If you want to practice this, let me pause for 1 second. If you want to practice it, I do recommend you can try this out if you go to Educational Games, NobelPrize.org, Educational Medicine, Blood Typing Game. Just do a Google search for Blood Typing Game. Make sure you guys go into the Educational Games, NobelPrize.org, and you can play this. It's a really interesting way of just seeing what you understand about blood typing and the antibodies present in the agglutination reaction. So, type A possesses A antigen and anti-B antibodies, so they can receive blood from themselves or O. B blood has B antigens and anti-A antibodies, can receive blood from itself, B, or O. AB has both A and B antigens but no antibodies, so it can receive from A, B, AB, and O. And O can only receive blood from O but donate to everyone, as you can see. Okay? Because it has both anti-A and anti-B antibodies, the only way to actually get... So this table also helps you guys do that. So with this table and this image, you should be able to answer the questions. There will be 3 questions about blood typing, whether it is about the antigens present, the antibodies, and who can give to who. Okay, that kind of stuff. So you really need to understand this. This is definitely going to be a higher order question on your second exam. Okay. Now, these are not on the exam, but I do want to make sure students understand that between different races and ethnicities of humans, there are different distributions of blood types, where certain antigens are more common in certain populations. For example, in America, You might see that, for example, percentage 100% of Chinese Americans have the Rh antigen. Things like that. So this is why when you guys see you might see the Red Cross or our local blood bank, which I just forgot the name of, Marsh might say, we need more African American people to donate blood, or we need more people of Filipino descent, things like that to donate. It's because different blood types are distributed differently amongst different ethnicities around the world. So that's very interesting and actually quite important. Now, normally this is not a fatal thing with an Rh incompatibility. Is not something you have to worry about unless you've been sensitized to it. You've gotten it. So that means you have to be an Rh negative individual who gets exposed to Rh positive, then you will make antibodies against it, and the second time you see Rh positive, you will attack it. Now, here's a problem though. In mama she gets exposed. If we have a mother who's Rh negative, she is pregnant. She was born, obviously. She gets pregnant with a child who's Rh positive. And this is her first child. That baby is fine. No problems. She will make anti-Rh antibodies. Why? Well, when she gives birth and the placenta hemorrhages and separates, mama's Rh negative blood mixes with baby's Rh positive blood, and she makes anti-Rh antibodies. Well, she's fine because she's Rh negative. It has no problem. And she never will change her blood type. But when she has a second baby who's Rh positive, antibodies can cross placenta. And when antibodies cross placenta, it will go in and kill the fetal blood cells, possessing it, and can kill the baby. Now, one of the things that happens is very common for babies who have either an A, B, or Rh incompatibility be born jaundicey. And then especially with, now A and B, they're not as serious. They usually give mild jaundice, baby's okay. It's the Rh one that's potentially fatal. It's called hemolytic disease of newborn, or erythroblastosis fetalis. I do have a question on erythroblastosis fetalis on my exam. I'm not going to tell you exactly what it is about it, but it is on the test about about this mechanism. So make sure you study it. Now, so as you can see, when mama gives birth and placental hemorrhaging happens, this starts to cause the blood to mix. Mama, after birth, has the anti-Rh antibodies, but antibodies can cross the placenta. And oftentimes the baby dies after birth. So this is why it's hemolytic disease of newborn, is the baby doesn't die in utero, it dies afterwards. So this is a cause of death. So RhoGAM is given to trick the mother to think she has made anti-Rh antibodies so that she doesn't make enough to make this a fatal thing. It's a little trick, and it's a product made from whole blood donations where the Rh antibodies are removed from the blood donated and then used as a medicine. So, but it is a whole blood thing, so you got to be, anything made with whole blood, there's some risk, so they have to scan it for HIV, things like that, hepatitis, any bloodborne illnesses, okay? Now, white blood cells. That's what we're going to next. White blood cells, just to remember, there are two types of white blood cells out there, and they fall in two categories, the granular ones and the agranular. Now, I'm just going to say this. First off, you're like, granular? What does it mean to be granular? Well, granule is, you remember in AP1 we talk about vesicles, lysosomes. Vesicles and lysosomes contain chemical compounds used in immune responses. They contain acids and enzymes, things that will harm pathogens, things like leukotrienes, prostaglandins, heparin, histamines, bactericidal compounds, and even acids, and other things will be in granules. And they're present in all white blood cells, but it is about when they're stained. This is important. Granular leukocytes contain stained granules. the granule state. Now remember, it's bin granular. Think of our white blood cell terrorist, bin granular. Bin granular is our granular leukocytes. Basophils, eosinophils, neutrophils. Agranular loves money. Lymphocytes, monocytes. I forgot to make the L and M bold. So granules are always present. It's just they may not appear with staining procedures, depending on the chemistry of that, how we stain it. Okay? So bin loves money. Bin, granular, loves money, agranular. Okay? Now, what we also need to understand is something called a differential count. And what a differential count is after I stain a white blood cell, I can then look at it under a slide, under a microscope, and qualify it. And then count them. So, how many basophils, eosinophils, neutrophils are there? How many lymphocytes and monocytes are there? And get a count. Now, the count is used to diagnose disease. So, we always do this on the staining and shapes, morphologies. We qualify them, and then we count them. So that's what's going on. Now, that is indicating a disease. For example, never let monkeys eat bananas. That is the appropriate differential count. I will talk about it. Your neutrophils, your lymphocytes, your monocytes, eosinophils, and basophils. For example, if there's not enough white blood cells, that the levels are too tinea, tinea white blood cells too few, leukopenia is when white blood cells are tinea. There's not as many. Leukocytosis, white blood cells are the mostest. There's too many. Leukocytosis, white blood cells the mostest. Leukopenia, the white blood cells are tinea in number. Okay, like leukemia, a leukocytosis could be leukemia. You have white blood cells that are overproducing, a type of cancer. Now, white blood cells, we're going to talk about, have a lot of things going on about them. And I keep meaning to fix this slide in a minute. It's phagocytes, NIM. But amoeboid movement is something they do. Now, amoebas, you got to think about an amoeba. An amoeba, what amoebas do is they ooze out and move. So they move around. They're amoeboid. They like pull themselves around. And so the type of mobility to attack a pathogen is called amoeboid movement. Now, another thing that white blood cells can do, normal cells don't do, is they can leave the bloodstream. They can leave capillaries and leave to go to the tissues. This is a fancy name, is diapedesis. Diapedesis is basically when a white blood cell yeets out of the capillaries and they go to your tissues. They just yeet their way on out. Okay, that's the white blood cell version of yeet. Is diapedesis. Now, also, they are attracted to chemical stimuli. That is called a positive chemotaxis. They are positively attracted towards chemical stimuli. They will see something going on, and they go towards it. For example, when a cell comes in contact with a pathogen, a disease-causing agent, they'll send out chemical distress signals, a chemical attractant. We're going to learn about them later on. And that attracts the white blood cells. It's like sharks with blood in the water. It's the cells that when white blood cells come in contact with the pathogen, they release an SOS that gets more white blood cells to come in, the ones that can deal with it, or other cells that come in contact with these, like a normal cell might release these things. We're going to talk about these are chemokines, and we'll talk about how the chemokines, when macrophages to the area, and they're like, I phagocytize you. Now, phagocytosis is something that also happens. The three phagocytics are NIM. Nimophagocytes. Let's just say NIMO, not NIMOLE. I don't know why this it's a really bad Mexican restaurant named NIMOLE, but nimophagocytes. The nimophagocytes, because we're from the South, and Mama Nims, so we NIM. That's them and them. So that's nimophagocytes. Nimophagocytes. Neutrophils, eosinophils, and monocytes are all phagocytotic. They're phagocytes. They can do phagocytosis of a bacterial cell, for example. Now, white blood cells, how do they... Now, they also roll on the surface. And what you'll see, this is on the capillary wall. You see me rolling, hating, waiting to diapedesis. So they're waiting to find a gap in the blood cell wall where that white blood cell will yeet diapedesis out. So he's rolling on the surface, waiting to find a hole where an injury might have happened so it can go out to the surrounding peripheral tissues. So the differential count, the numbers I will not test you on, but the order of abundance from most to least I will, and this is on the exam. Never let monkeys eat bananas. Neutrophils are the most abundant, somewhere between 50 to 70%. You're not going to get tested on the percentage. Lymphocytes, second most abundant, about 20 to 30. Monocytes, about 2 to 8, third most. Eosinophils, fourth most, about 1 to 4%. And basophils from anywhere from 0.5% to 1%. And they're at the bottom, so they're in the basement. So never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils, most to least. Okay? Abnormal, happy, healthy human. And this is something I'm going to test you on. Okay? Now, one of the other things I'm going to have on my exam is I'm going to ask you guys to tell me I'm going to give you guys a description of a white blood cell, and you're going to come in and I'm going to ask you, what is it that the white blood cell, which white blood cell fits this description? Okay? What does it mean? I might say, which of these white blood cells indicate person is infected with worms? Something like that, okay? So the neutrophil, which is the most abundant, never let monkeys eat bananas, never, most abundant, 50 to 70, are called polymorphonuclear leukocytes. Now what does that mean? Here is its nucleus, and notice it's got all these weird shapes. It's got a nucleus with many morphs, polymorphonuclear. It has somewhere between 2 to 5 lobes. This one has 3 lobes. They could have 2 lobes, 5 lobes, 4, 3, okay, anywhere in between. They are granular. Here's the granules. You can see their little granules all scattered around. They are granular, so bin granular. These granules have lysosomal enzymes. Some of them are lysosomal enzymes. Those are carbohydratases, lipases, nucleases, proteases, and acids. Also other bactericidal compounds. We're going to talk about some of those in the immune system. They are also first to attack. Neutrophils help neutralize because they're the first to attack and phagocytize, and they have to do that when there's an antibody present. So like with a red blood cell, we saw that we destroyed those red blood cells because the antibody attack was on it. And this is kind of, think about an antibody is like a mobster putting a hit on a gangster. So when you release the granules, this is called degranulation. It releases these things. And it releases prostaglandins. Prostaglandins, remember, is for pain and inflammation, type of lipid. Prostaglandins, for example, is somebody were to visit a prostitute, they may end up with pain and inflammation after. And then they also release leukotrienes. Leukotrienes, these are helping to control spread of infection and inflammation. So this indicates an infection, usually a bacterial infection for a neutrophil. Neutrophil usually tells you there's a bacterial infection. Okay? Lymphocytes, never let. Lymphocytes are second most. They have a naked nucleus. They're very large and much bigger than a red blood cell. Here's a red blood cell. Look how much bigger he is. He has this big nucleus with a thin layer of cytoplasm around it, which means he has that naked nucleus. He is agranular because, well, it has some speckles, but these granules don't really go out. Now, there are 3 types of lymphocytes: T cells, B cells, natural killers. T cells do cell-mediated immunity to attack a cell who's infected. B cells do humoral immunity. They release antibodies into the body's fluids to attack antigens present. These are the cells, B cells, that are making your anti-Rh antibodies. And there's the natural killers. They're present and kill abnormal cells and tissues like cancer cells. And they indicate an infection or maybe even cancer. Okay, I should put infection or cancer. So they indicate an infection, things like that. Monocytes. So most of them will indicate infections. There's a couple who indicate very specific things, like neutrophils specifically for bacterial infections. Monocytes. Monocytes are 2 times bigger than a red blood cell. So they're massive monocytes. Monocytes are massive. They are never let monkeys, third most abundant, between 2 to 8% roughly. Now, they go around through the bloodstream, and they are massive, and they become the massive macrophage, the big eater. Macrophages are large phagocytes, so they phagocytize. Phagocytes. They have this bean-shaped or heart-shaped nucleus to them. They act as an antigen-presenting cell. They are going to be what is needed to make sure that a T cell can recognize an antigen and help antibodies to be produced. So they also indicate infections. All of these do, but these guys are an antigen-presenting cell. That's the most unique thing about them. And they become macrophages. Now, eosinophils, they look like Spider-Man. They're called acidophils because they take up eosin, who is acidic red stain, but that's why they're called eosinophils. They love philos, eosin, eosinophil. They look like Spider-Man here, okay? They have deep red granules in a bilobe nucleus that makes it look like Spider-Man. So they are granular, been granular. They always attack things with antibodies. So that also is for parasites like worms. So this might indicate a worm parasitic infection. Now, they release toxic things to do that, but they also are activated by allergies. Eosinophil. They control the spread of inflammation. Since they control inflammation spread, try to prevent inflammation from going all over the body, they're easy on you, phils. Basophils, the lowest number, are very dark and basic. They take up a dark blue basic stain, and they are extremely granular. They're so granular. How granular are they? They're so granular you can barely see the nucleus. And the granules contain histamine and heparin here, and they help me histamine to dilate blood vessels to bring nutrients and more blood, white blood cells to an infected area, and heparin to prevent a blood clot to that area so that you can bring lots of good helpers to fight things. So they also cause inflammation. So they are allergic reactions. They cause inflammation. They stimulate it. So they're bad on you, phils. Basophils are bad on you, phils. All right. So please make sure you pay attention to a few of the indicators. So especially parasitic infections, cancer, and bacterial infections. Okay. So platelets or thrombocytes, they are these disc-like fragments, and there's enzymes to help favor clotting. They don't have nucleus, but they make these enzymes to help us make proteins. There's a cell called a megakaryocyte, and the megakaryocyte's massive. Then he breaks up into many pieces, and these little pieces, when they're activated, are called a platelet. So that's why we call them cell fragments and not platelets. Now, these platelets, there are about 2/3 are in the blood, and a remaining third is in your spleen. If you get hurt, your spleen can knock it out. To stimulate them, we learned that erythropoietin made red blood cells. Well, thrombopoietin causes platelets to form, and they cause a blood clot. Now, for example, thrombopenia is when you have low numbers of platelets. When thrombocytes are tenia, you have thrombopenia. When thrombocytes are the mostest, you have thrombocytosis. So low numbers can cause excessive and unwanted bleeds, where thrombocytosis actually causes unwanted clots. Both of those can kill you. Now, platelets like to party, and there ain't no party like a platelet party because they aggregate adhesive and they agglutinate. Platelets like to gather together, stick to damaged surfaces, and clump together. So they gather, they stick, and they clump. They aggregate, adhere, and agglutinate. They gather together, they stick to a damaged surface, and then they clump together to seal a wound. Okay? That's how they do their job. Aggregation, adhesiveness, agglutination. Okay? So here's all the formed elements. You can see kind of their basic characteristics and things like that. This will not be on the test other than the myeloid stem cells are involved in everything but lymphocyte production, and that's a lymphoid stem cell. I used to test on these, but we now know this is not right. This is not how this works now. Our book is not caught up to the latest research that's come out saying that this needs to be redone. So I'm not going to have you memorize something that's no longer correct now. I've put it in there if you ever need it, but let me just say students only need to know what cells have myeloid or lymphoid stem cells. Okay? Now, colony-stimulating factors cause white blood cells. For example, if it's the monocytes or called the MCSF, the monocyte colony-stimulating factor, and even the granulocytes is the granulocyte colony-stimulating factors, things like that. All right, but all you need to know from this slide is who has a myeloid, who has a lymphoid. Okay. Now, I urge you kind of pause here and kind of come back a little bit because hemostasis is going to be a big one. And then after that, we really are done. But this is the last big thing. Blood typing and hemostasis are the two hardest things. So if you did stop and break, let's jump on in now. Okay, so what is hemostasis? The stopping of bleeding. What's the fancy word for stopping? Cessation. Cease and desist. Stop bleeding. Hemostasis. Hemo, blood. Stasis, stopping. Stopping bleeding. Okay? Hemostasis. There are 3 stages for hemostasis. Vascular phase, platelet phase, coagulation phase. Vascular phase is when blood vessels will spasm down. Platelet phase, platelets are going to start to stick together, do their platelet parties. In coagulation, we're going to start turning plasma into a thick jelly. Okay? So let's talk about this. Now, first, before we do anything, it's important that we know 2 things that almost everything in clotting needs is calcium and vitamin K. Vitamin K, for example, if you don't get it, is why they give babies vitamin K shots after birth. It is common for them, if they don't get, people say, I'm not giving my baby a shot, I'm not putting things in my baby's bloodstream, and the baby gets an injury and they die, they bleed to death because they will not be able to clot. And calcium is needed for blood clotting. Calcium becomes a cofactor for making most of the proteins to link together to actually form fibrin strands. The other things needed is proteins called fibrin and gin, and all the enzymes that are coagulating enzymes. You know, the factor 1, 2, 3, 4, 5, 6, 7, 8, 9, for example, factor X. All those clotting factors, factor 13, all those, right? So those enzymes, coagulating enzymes. Okay, so what is the vascular phase? If I get a cut, for about 30 minutes, a blood vessel is going to spasm down, get smaller, and trying to reduce blood loss. But when that happens and the cells contract, as that happens, you will get the endothelial cells exposed. They contract as well. When the blood vessels contract, the endothelium shrinks up, the lining of a blood vessel. It shrinks and exposes the basal lamina. And that basal lamina can actually act as an activator for platelets we're going to see. Now, the damaged cells that line a blood vessel called endothelium, they release chemical factors. They release adenosine diphosphate, ADP, a tissue factor, and PGI2. These are chemical factors to activate platelets. And then the endothelins will cause the muscles to contract, vascular spasm, maintaining that spasm, and cause cell division to start to make it want to heal. And lastly, the endothelial cells get sticky. And that helps see all blood flow, but it's going to get sticky for another reason. Now, this stickiness, I'm going to say why it allows platelets to adhere to it. Now, if you go into healthcare, these endothelial cells, when they're damaged, they produce a molecule called von Willebrand factor. And you probably will hear about this in healthcare programs, is von Willebrand factor. That's when it gets sticky, that's what it's making. It's called von Willebrand factor, and it makes like a hook for a platelet to come in, and when the platelet who's been activated by ADP, tissue factors, and PGI2, they will come in and grab and be snagged by the sticky endothelium, the von Willebrand factor, and they will make it stick to the damaged surfaces. So will the exposed basal lamina do the same thing. So this all sets the stage for platelet phase. Let me show you. When the collagen here, when collagen from the damaged blood vessel is sticking out, a platelet can get activated. So he's got to be activated. So platelets, to be activated, there are three different things that happen. The platelets can get, when they're activated, they can stick to the sticky endothelium. That is what is referred to as, that's what von Willebrand factor does. So if you have von Willebrand disease, it's going to make it harder to do platelet adhesion. Platelets then start to stick to other platelets, because platelets, when they're activated, they will start to make themselves sticky and make other platelets adhere to them. And this starts to close small breaks. It starts to form a platelet plug. Okay? And that platelet plug will hopefully fill this wound. Now, the platelets get activated by all these chemicals like ADP, the PG, the platelet-derived growth factor, calcium released from other platelets, clotting factors, serotonin can help cause the muscles to contract, because that's one of the things that an activated platelet releases is serotonin. But they release what's called PDGF from activated platelets, calcium, clotting factors like thrombin, and ADP. And the ADP, imagine right here, you have a platelet coming through, and he's very sleepy platelet, and he hits ADP, and ADP is like a cup of coffee for a sleepy platelet, and that helps activate him. Calcium helps activate him. Clotting factors help to activate to make the platelets stick to each other. Okay? And the clotting factors will also activate the next phase called coagulation phase. Okay? So now we formed potentially a platelet plug to prevent the thing. Now, platelet plug, we don't want the whole body to turn into one giant clot. So there is a prostacyclin, which is a type of prostaglandin called prostacyclin, PGI2, that inhibits the amount of platelets. So it becomes inhibitory from your white blood cells to help keep the platelet from spreading too much. You don't want the whole body to turn into one clot when you get hurt, because if I just do that, I probably damage 15 capillaries, 100 capillaries just from doing that, okay? And if I did that one time, my whole body can turn to a clot and I die. So it's important that we inhibit this. And there's a reason I'm teaching this. Trust me, you need to know it when you get to your programs, okay? So the ADP acts like coffee for a platelet who's sleepy, and he comes by and he wakes up with the coffee, the ADP. Wow, I'm awake now. Thrombin also gets him going, and the exposed collagen fibers of the damaged vessels here, they will help to do this. And that is going to activate these platelets to release clotting factors, and these clotting factors will set up coagulation cascade. Okay? And that's where we're going now. Is coagulation. Now, coagulation begins about 30 seconds after injury. So, you got that vascular spasm is happening for like 30 minutes. So, even after 30 seconds, coagulation starts happening. Now, a lot of people want to take clot and coagulation and make them the same. They're not. Now, a platelet plug, think of that as a clot. Coagulation reinforces that. Okay? Now, what we're going to do is there's two ways to do this, but there's two ways to kick off the main coagulation cascade. But let's just get it and step back for a minute and say, what is coagulation? All it is I want to take this fibrin proteins that are dissolved in blood, soluble fibrinogen that's soluble, and turn it into fibrin in the blood that is not soluble, and reinforce my platelet plug with it. To reinforce it and make that a stronger clot. Now, if you guys go into healthcare, you'll learn the difference between what's called a primary platelet plug, which is what this right here is. This is a primary platelet plug. This is a secondary platelet plug. If you're in healthcare, you'll have to be able to discern between primary versus secondary platelet plug. Now, to do this, there are two pathways to cause coagulation. One is called the extrinsic, and that's external trauma, like a punch to my face, and you give me a black eye. And the trauma damages my blood vessels from the outside, opening them up. That will stimulate, that tissue damage will stimulate this avenue called the extrinsic pathway. Intrinsic pathway is internal damage. What would that be? Let's say you were doing a cannulus, or you were doing a catheterization, and the tube, as it went in, scraped the blood vessel's wall and removed the lining cells, the simple squamous endothelium here. That would open up the clotting pathway as well. So like a cannulus, a line being put into a blood vessel, things like that, okay? And this would initiate the intrinsic, internal damage. Both of these initiate the common pathway, but the extrinsic is faster to activate. It's easier to activate the extrinsic. The intrinsic is the very hard one, the really complicated one, that has all the really complex pathways to it, that if you ever have to learn, is the ones that are really, really, really annoying, okay? It's the ones that have all the different factors, the big cascade. But they are both important to activate it and act as a positive feedback loop to reinforce. So the intrinsic is very reinforcing. But the goal is to come from the extrinsic and the intrinsic to activate factor X. And when factor X is activated, then we can activate the rest of this. Now, what is happening here? Okay, I'm going to try this for the first time like this. In plasma, we have a material here which is a proenzyme that is present. So when you see pro something, we know it's before. This guy here, we need to take a material called prothrombin and convert that to thrombin. Now, how do we do that? Well, we need an enzyme here to turn prothrombin to thrombin. That enzyme that cleaves this is called prothrombinase. And prothrombinase turns prothrombin to thrombin. Now, thrombin is an enzyme that comes in and catalyzes another reaction. It turns fibrinogen, and pro, tell me these are inactive, to fibrin. Now, fibrinogen is soluble in plasma. Fibrin is insoluble. So what you get is all these little fibrin strands. So basically, a little fibrinogen is like this little piece right here with this little inactive thing on it. And when thrombin comes in, it removes it. That releases, and I can take another activated fibrin and keep chaining them together till I make strands of fibrin that can come in and reinforce the clot. Okay? Now, so prothrombinase activates prothrombin to thrombin. Thrombin activates fibrinogen to fibrin. And fibrin, this is the clotted blood, the common pathway. I will have this on the test where I ask you what does what in the common pathway. In the common pathway, I might say, what converts prothrombin to thrombin? Prothrombinase. What converts fibrinogen to fibrin? Thrombin. Okay? Now, thrombin also activates platelets. So, thrombin can be made here by this cascade or release from an activated platelet to activate more platelets. And that thrombin can also be brought in. You can just bring some thrombin from platelets being secreted, and that can be brought in, reinforcing the production of this. So I can make a little bit of fibrin or a whole lot of fibrin and everywhere in between depending on the severity of the damage. This is the importance of the coagulation cascade. Okay? Now, the last thing I do want to talk about here is that the clot that we've made, the platelets can get pulled off after it takes about 30 to 60 minutes. You can start getting this. Now, the thrombin, not only does thrombin activate platelets, not only does thrombin convert fibrinogen to fibrin, but thrombin also activates plasminogen. Plasminogen gets turned into plasmin. Plasmin dissolves the clot and digests your fibrin strands and activates this. So to activate plasmin, you need a thing called tissue plasminogen activator. So tissue plasminogen activator, or TPA, will activate plasminogen into plasmin to dissolve that. TPA is called a clot buster, and this is needed for stroke patient. You've got a short amount of time to give a stroke patient the clot buster to block that, or somebody who has a clot, that like an embolus or any kind of thrombus going through the bloodstream. You can break it up and prevent the stroke, but you have a very short time, or a pulmonary embolism, something like that. A clot that wanders through the bloodstream. Okay? So TPA, tissue plasminogen activator, activates plasminogen to make plasmin dissolve the clot. That's why it's a clot buster. Okay? What are the 5 things that inhibit clots? There's an anticoagulant called antithrombin 3. Antithrombin 3 actually inhibits thrombin. And heparin sulfate is what stimulates antithrombin 3. So heparin, things like that, can stimulate the antithrombin 3 to inhibit thrombin. And inhibiting thrombin. Remember, thrombin converts fibrinogen to fibrin. If you don't do that, then so this can act as a feedback loop here. So heparin also prevents clots. Heparin prevents clots. Heparin. Thrombomodulin. Thrombomodulin, what it does is there's a protein called protein C that it activates activates the clotting factors, so it acts as a clotting inhibitor. And then TPA, clot buster. So, and then prostacyclin inhibits platelet activation. Prostacyclin is PGI2. It's a prostaglandin and can prevent you from inhibiting platelets from being activated. So it can keep them sleepy. Plasminogen, so plasminogen activators cause plasmin to be broken in, so you get fibrinolysis and you break the clot down. So here's some diseases not on the test. I also, just for later, here's a list of all 13 blood clotting factors from the coagulation cascade for those of you who had to ever learn it. This will be useful for you, okay? And that's it. So the big things here, guys, I really want you to pay attention to is the common pathway and the different stages of the clotting phase. Make sure you do understand that it is a very big deal for you guys when you go into programs. I mean, honestly, I can tell you from experience, you're going to need to know it, and people are not going to teach it very much or in very much detail, and the detail students need. So, as I said, through every chapter, make sure you read the chapter objectives. Those are what I base my test off. So, guys, this concludes the end of chapter 19. Be checking out for the lab lecture as well. So this new thing here that I'm using can actually allow me to be able to put the models under now. It's a nice little camera that I have. It's an 8K camera, actually, so it's pretty cool. All right, guys, this concludes my video. Thank you so much.